everyone. Hello, everyone. I'll be introducing Dr. Contreras. Dr. Contreras is a professor of chemical engineering at the University of Texas at Austin and currently serves part-time as an associate dean for postdoctoral affairs within the graduate school. She is also a member of the microbiology, cell molecular biology, and biochemistry chemistry graduate programs. And she teaches Introduction to Chemical Engineering Computing, Thermodynamics, Intro to Chemical Engineering Analysis, and Fundamental and Applications of Cellular Regulation. I'll leave the rest to her.
Okay, so I am welcome everyone. I am glad to that you're logging in today on a Friday afternoon to spend a little bit of time, I guess, getting to know a little bit about my trajectory, as well as a little bit about what our group is working on science. And of course, I think as you realize today, those two things are interjected. And so I wanted to start by telling the audience a little bit about myself. I was born in the Dominican Republic. So I was there until I was about 10 years old. And then I immigrated with my family to New York City. So I am the child of immigrants. So by that definition, although my parents were not the first generation college people, they went to school in a completely different country. And I am the first in my family to really pursue a career in STEM. So I guess in many ways that makes me a first-gen person in STEM. And I currently live in Texas, so I am at UT Austin, where I started my career in chemical engineering with links to cell molecular biology, microbiology, and biochemistry about 15 years ago. So I have mentored a number of students. I have built a great team. And our research, as you'll see today, focuses on basic innovations, understanding, creating systems to study fundamental interactions in regulation between proteins and RNA, which gives us a lot of freedom to explore a lot of problems. And one of the ones that I'll be talking to you about today, it's something that I've been very interested in recent times in my career related to air pollution. And so, as it was said during the nice introduction from Christina, I also wear multiple hats, multiple roles. So in addition to being a professor in chemical engineering, I have held other university level administrative roles, including vice provost for faculty affairs, where I've had to think a lot about what does it take to build an inclusive campus? What tools do our faculty need to make sure our students are feeling that and experiencing that, in addition to how do we welcome everybody and make sure we cultivate everybody's talent on our campus. Most recently, I have been an inaugural dean of postdoctoral studies. So in that role, I am responsible for really thinking a lot about grad students and postdocs and what do we need? What do we need to really transition to a culture where we provide people what they need to be the best that themselves can be? So what do we need to bring that out? What are programs that can really engage everybody and provide that level of training so that our postdocs in particular and our grad students can really go and embark on any career that they want out after being here at UT Austin. Other interesting things that also I have found around my journey that have really provided a way by which I've been able to use science and STEM to serve different communities is that I have also had the privilege of, alongside the Innovation Center here at UT Austin, of being a co-founder of a startup that's really using technologies and things we've patented to really serve families in need through a company called Griot Baby, focusing on baby products. And as a community member here in Austin, I also have had the opportunity to serve and lead in a couple of nonprofit organization boards. One of them is Hand to Hold, which is an organization that serves families that are bereaved because they have lost a baby upon birth, or because they've had to go to the neonatal intensive care unit with babies that are usually tiny, tiny babies, sometimes a pound, sometimes 2 pounds, so tiny, tiny babies. So I have had also the opportunity to work on and use my STEM training to really be able to bridge communications between underserved populations and sort of the science that doctors often communicate in hospitals that often makes a lot of people feel left out or not included in how they understand these medical processes in the hospital. So I say that because I want to start by stressing to you all that when I embarked in science, I remember being at Princeton thinking through, I really want to be a doctor because that's the only thing, a medical doctor, that I recognize as a profession that had enough ties and enough meaning in that community to help people. So perhaps given my background, perhaps because I grew up around a community that was really underserved, when I arrived in the country, for me, it was very important to find a profession and a discipline that allowed that personal interaction and that allow that personal giving back in ways that meaningfully touch people's lives. And the stereotype that I heard of science and STEM in my mind, and the way I was always presented in the little bit I knew about it, because as I said earlier, I didn't really know anybody personally that was an engineer, a chemist, a biochemist, a physicist. I had a hard time initially realizing how anybody in STEM could really have those ties to their community. I mean, what did these people do? It was very hard to get excited about it. And so I've been very privileged throughout my career that I've been able to use a lot of the skill sets that we're all learning as grad students, postdocs, professor, to really benefit communities that I work with. So that has been very important throughout my career. So I wanted to touch on a couple of things today that I always get asked about that I think would be useful as you're all thinking of your own journeys, careers, and what do you do afterwards? How do you make all this work between what you're passionate about professionally and maybe what your personal passions can be? So one thing that I usually get asked about is, when did you know you wanted to be a scientist? So, right, the theme is always, when did you figure out and how do you keep that going? How do you keep that going with all the things that we encounter every day, with all the pressures that we had every day? And so I kind of knew from a young age that I love math and science. I really wanted, as I said, something that helped people. Eventually, I became in love with biotech because somebody gave me one opportunity to do one visit in one of these kind of summer programs. So that in retrospect really has fueled a lot of my excitement about community outreach and a lot of the work that we think of doing when we go into those middle school classrooms or talk to teachers in grade school. For kids like me, that was pretty much my one window into what the world of STEM could look like. I remember looking at a petri dish in one of those visits by a grad student back in sixth grade, and I was fascinated. So that really turned my interest into starting my path in STEM. I was also pre-med when I was at Princeton as an undergrad. And so again, that's kind of all I knew. So I kept that alive because that's what I really thought I was going to go. But the more that I got into research, a professor gave me an opportunity to be an undergraduate research at Princeton. So I really got to figure out, what do researchers do? And I fell in love with this idea that you were there discovering, learning, maybe something that people didn't know. Maybe you were the only person in the world or a few of those that were studying this one topic. And so that's how I really became very interested in RNA, in protein engineering, and got me to go to grad school. So then a lot of people ask me, well, you went to grad school, you decided to go as path of your journey and sort of pursue this STEM thing. What was it like to be, right, underrepresented as a woman in that field? What was it like to be underrepresented as a first gen person in that field, as an immigrant, as an international background person? And as you all know, in the field of engineering and most of STEM, women are still about 30% in a survey from 2019 or so of represented in the field. And so when you think about what that really means, it gets smaller and smaller. This is like in the profession as a whole. So that includes undergraduate degrees, that includes the workforce. So if you think about where we all are, that even gets smaller as we keep going up to grad school, postdoc, through the professoriate. And if we add all these other interesting factors about ourselves, ethnic, racial, socioeconomic backgrounds, I'm sure the numbers grow up pretty little. So that's a kind of a statistic that is inspiring. And we also have to recognize that that presents an interesting situation. And for me, how it affected me most was in essentially the dilution of role models. It was very hard to figure out as a grad student, who do I emulate here? Who do I want to be like? Who's kind of like me? Who approaches things in a way that makes sense to me, in a way that they approach relationships in a way like I do, like I grew up to do. And so I think that conflict that it presents when we're trying to find a way and we're specializing more and more in grad school comes down to the question of how do I reconcile in this field as a STEM person, whoever I am, what's valuable to me? How do I reconcile that? How do I figure out how to do this? And so I think I can give you countless of examples where this came up, but I distinctly remember when I was about to go and find a job, an interview for positions as a faculty member. You might laugh, you might think this is silly, but I struggled for weeks figuring out what do I wear for an interview, right? Everybody that I've never seen somebody wear a dress. I've never seen somebody wear a skirt, but that's kind of how I feel dressed up. That's how I feel professional. So conflicts like this, another big one came up when I became a mom. I was already a professor and I was actually breastfeeding. So I had to go and pump in the road while giving seminars. And I remember the conflict of, what do I tell people? Do I tell them that I need time on my schedule and a room because I'm sick? Do I tell them I need to rest? So it's this conflict with not revealing things about yourself because you've never seen how this is done. You never know how this can affect your trajectory, perception of people, how people will receive it. And it's just not the norm. It's just not what happens every day in these environments. So you feel isolated, you feel sort of alone, you feel that there's nobody else like you. And I think that those examples are very women-centric, but I know that a lot of you can find instances, whether it's because of your beliefs, who you are, how you approach things, your priorities. Maybe you really see your career much more balanced than what your advisor does and what the field communicates. And you feel embarrassed to bring that part of you that, you know what, I really like biking on Friday at, 6 o'clock. And so how do I communicate that when I feel this pressure to do, to be in the lab? How do I still remain somebody that's taken seriously if I fully come out how I am? So I think that's a theme that resonates a lot with me because of my own personal experiences that are, challenging when we are really looking at STEM still as such a monolithic and still discipline where we still see very little diversity in terms of the models that are out there that we really call successful. And so I've been really fortunate to find, what you're doing, finding people in grad school, keeping these cohorts. That has been great for me to reach out to my grad school friends, my postdoc friends, where we can really have these conversations and support each other. Because the reality is, the good news is things are changing. And a lot of this sort of group of people that are wanting to embrace STEM are realizing as a culture, we have a lot of work to do, but we can support each other. So another theme that I wanted to touch on was this idea of how do you deal then with a lot of these stereotypes that you encounter in grad school, a lot of these things that you feel are, maybe you get really frustrated because you know that, you may not be given the best projects or the best opportunities, so people may not be thinking of you at first. And I think that that's part of a lot of this theme of the culture still has a way to go. But I think it's really changing. So if you're feeling at points like you really have to be much more effort for you to raise your hand and say, hey, here I am. I feel not seen. I feel ignored. I think a lot of those things are real. I think they're there. But I think it's part of a broader culture that we're all invested in trying to adapt so that, there's inclusion really in all of our disciplines. So I think we all have work to do there. But that's another theme that always comes up when I talk to early career STEM trainees about the field, about what we're doing, about things that affect them every day. So one of the things that usually comes up, and I tell them, is, look, this is not just even, your program or my program. This is a big, deep societal issue. I still remember when I was a little girl, I would get all the dolls to play with. And they were so boring. They didn't do anything interesting. But my brother got all the Legos and all the cars that made noises and could be remote control and planes that could fly. So from a very early on, this idea that we have been and we keep kind of boxing people into what they should or should be interested in, I think continues to be a challenge that is one that we keep encountering as we go deeper into our training. And so I think that those views that we get where people seem to box us as, in the case of women, a lot of times it's as caretakers and nurturers and the people that should really carry the room. That is kind of poses a lot of problems for us sometimes, because science and STEM seem as very quantitative and it seem as very sort of one answer, not a touchy-feely thing like people mostly associate with a lot of gender stereotypes that are there. So those things are pretty real. And again, it's great that you all have this community where you're talking about things, where you're finding people that you can talk about these things. Because to kind of end this little bit of an introduction about my path and things that I've faced and continue to sort of work through, I think we have a lot of tools, a lot of people, a lot of structures emerging in place that have recognized that we all can bring something to STEM. We all can succeed. I acknowledge at times we feel discouraged. We feel that, maybe the path could be a little easier. But I think it's really encouraging to see how many amazing people of all backgrounds are coming into STEM and really changing the discourse of who is a successful engineer, scientist, etc. And I think as we continue to see that positive energy, that excitement, people like you all sitting at a table, recognizing that, these challenges are still out there and we all have to do our piece. I am very hopeful that we're going to continue to see a lot of changes in how the discipline evolves to welcome everybody's needs, to welcome all the talents in there, and that is quickly changing. And so that kind of summarizes my interest, how I felt into this. And I will pause here, I think, and see if you guys have any more questions that are not outside or more that you'd like to hear about this first part of my introduction to who I am as a person before I switch to Lydia, the scientist. Okay, well, maybe I will switch over and we can talk a little bit about some of the work that my lab does that I am very excited about. So I think I need to share. Actually, I shared in the wrong way. I'm going to try it again. Okay. Sorry, guys. This is not the right presentation. I apologize. Let me try this again. Close this one and try sharing again. All right. So I just want to tell you for the second half a little bit here of what my research group is interested in, one of the recent problems that we're working on. And I hope you can see a little bit about how this is one problem that I am particularly excited because I get to use some of my strengths as somebody that is in love with RNA biochemistry and molecular engineering and systems biology to really look at a problem that affects a lot of people and that it's becoming pretty timely in how we talk about environmental equity, health equity, et cetera, in our communities. And so, right, so air pollution has been associated with many underlying diseases. As I'm sure you know, just the other day, last week, there was a headline in the New York Times about how it affects brain diseases. So we heard it in terms of respiratory diseases, cardiovascular diseases, more recently, brain disorders like Alzheimer's, Parkinson's, strokes, etc. And what we know now is that this could be a very real effect that affects millions of people with premature deaths every year. Now, what is really interesting about this problem is that it affects most and more deeply poor and racially ethnic underrepresented communities. So, for example, this is a courtesy slide of one of my pediatrician colleagues here in the medical school. And one of the things that we know is that if you go and you track asthma, this is data from the city of Baltimore, and you look at different demographics, they are demographics that are much higher incidence that correspond to low-income neighborhoods where asthma is much more prevalent. We also know that air pollution is extremely complex. And we also know that it varies quite a lot, these sources of air pollution based on geographical areas. So this is a model of our own neighborhood where you can see black carbon, a major pollutant, and sulfate, another major pollutant. And you can look at the city of Austin. The city here is bounded by this black thick line. And you can see the high concentration here on this part of the city that is very different from the rest. So when we're thinking about where different restaurants are being placed, traffic, metal factories, etc., it's interesting and important to keep in mind that that has a big effect on these communities. And so these sources vary a lot, and they contribute significantly to the particulate matter that we're seeing out there and how we can track medical data for its residents. So where I became really interested in this problem is that we know that a lot of what happens in the air is air chemistry. So essentially, it's a chemistry problem. And there is an intersection between that chemistry that happens out there and how living systems, the effect that it has on living systems. So how does this get deposited? How does it give rise to reactive oxygen species in cells, in whole systems like lungs and brains, etc. And ultimately, how it leads to biomolecular damage, which is really what I'm interested in, since I am really have vested interest in RNA, in proteins, and in regulation in individual cells. So just so that we're on the same page, PM reflects to particulate matter, and it varies based on chemicals and size. So you can have usually things that are at the micron scale. The smaller they are, the more they can penetrate. And an example of this is black carbon. So these are complex particles and a distribution of size and chemicals. So as I said, there's different sizes. The smaller they got, the more they can penetrate our bodies. And one of the things that we know is that we can assess these at different levels. So there has been for a while emphasis on systems, so whole organs, and then molecular pathways, and then all the way down to molecules, etc. And so there's a lot of scales that people are studying these effects, including community scales, so demographics, how the more like the data I just showed you, where people correlate exposure sources and sort of outcomes, medical outcomes. There's also a scale of the body, so organs and whatnot. And then there's the cellular scale, which is really our interest and the lead study. Within that cellular scale, there's a lot of emphasis in, hey, how can we say and how can we tell that there is an effect happening in these cells? So there's a number of biomarkers of air pollution that people have looked at for years. These include things like interleukins. So a whole suite of them that are being measured to realize, is there anything that is being responsive in these cells? Chemokines, inflammation markers, oxidative stress markers, etc. So when we started this work years ago, we were really interested in asking the question of, can we do a much more comprehensive evaluation on the effects of air pollution of lung cells through tracking different cellular property? So how do we evaluate risks? Are there more sensitive markers? And how can we track here tools, new tools, so that we can more readily understand these effects? So what I'll tell you briefly today, in the remaining of time we have together, is two things that my lab has been really interested in. Is in, can we engineer tools to really use these, focus on these cellular features and how they respond to this level of environmental stress? And all the way at the level of the RNA, what do we know that we can exploit for these models? So model systems that we started this work with have been epithelial lung cells. These are cells that are models, so they are in a data bank, they're reliable, they grow predictably, and they are in the epithelial part of the lungs. So this is the first barrier that the lungs gets exposed to when it gets exposed to any environmental threats or any environmental compounds. These are primary cell lines, so they are, we've used a combination of both model and primary cell lines. And what we have developed is a chamber here, one of the few now in the country, where we can collect exposures. Often, actually, we go to Houston with collaborators here that work on air pollution, and they bring samples and they analyze it. And our collaborators say, well, these are key compositions of this air that we have determined. We reproduce it in the lab, and we have a chamber that's huge, mixes it up. We create this air, we create chambers like this, where we can then expose cells to these mixtures, and some that are controlled, non-exposed. And what's really exciting about this system is that the cells sit at the air-liquid interface. So they're not submerging liquid, which makes it for a pretty relevant system that brings a lot of the properties that we can find in the lungs when we kind of pass these pollutant mixtures through for exposures. So using these, we have characterized a number of mixtures from sort of that contain urban type of settings, so cities like industrialized cities that we find. We have characterized samples that are more from industrial diesel-like exhaust mixtures, and also from other places that contain more of these fine atmospheric PM types. So different types of particles here. One of the big differences that we are finding is that a lot of these sources vary a lot in their metal composition. So in the example that I'll be speaking with you about today, one of the things that we realized is that lead and cadmium, for example, vary a lot by mass in all of these three types of samples. And so what we can do here is we actually, one of the exciting pieces has been that we can actually bring filters to different neighborhoods and use different grad students' homes and friends of the grad students, and we can start sampling. So this is our own city of Austin. These are 4 points that we sampled mixture from, and we collect them in this filter. We then do this type of analysis to measure the amount of PM by mass, so gravimetric analysis, and to do an elemental composition. And you can see these are various elements. They contain metals, they contain things like aluminum and silica, etc. And this is a relative proportion of them. And I know you won't get details, but what I want you to quickly see is that, again, depending on where you live, depending on where you are, your exposure and your type of PM that you're exposed to really varies per these analysis. So then we take these filters and then we sort of dissolved these PM here, and we exposed ourselves to them. And one of the things that we started seeing right away is that even if we look at the level of the cell, there's a lot of morphological remodeling that's happening. So what do I mean by that? It's very easy to see, even if we stain these cells, that if you compare clean air cells, these are lung cell models, to cells that are exposed to some of these pollutant mixtures, you can clearly see here that when you look at the red here, which kind of stains the cytoskeletal of these cells, you start seeing that the cells are less connected. You start seeing that they're losing connectivity. And if you focus on the nucleus, you can even see that the nucleus is starting to get misshaped. You can also see that shapes of the cells are kind of changing, getting distorted. So this is something that has been clinically sort of observed in many of these diseases, but we have been really interested in understanding a molecular basis of what leads to this over the years. So one of the questions that we've been asking is, do these different shapes correspond to specific concentrations or types of pollutions? Is there a threshold for any of these that induce these phenotypes? And what underlying biological changes are driving this? Can we come up with high throughput ways so that we could really empower people with new technologies so that they can understand their own risks to the different pollutions that they're experiencing in their communities and neighborhoods? So those have been really the motivating questions for this study. So I just want to show you a little bit of data so that you get a sense of where we are and how we're using our tools to answer some of this. So one of the first things we can do is we can do biochemical assays to measure cell viability. This is sort of a metric that lets us know how sick do these cells get in the presence of these pollutants, how dead are these populations, how toxic are these mixtures. And you can see, interestingly, these diesel, even at high concentrations, have very minimal effect to some of these urban concentrations that quickly drop cell viability. They're a lot more toxic. So we're starting to understand the importance of really characterizing these mixtures and really starting to identify what are key drivers of pathogenesis, what are key drivers of cells starting to not operate in their healthy mode after exposures. So this is where you can see, this is the percentage of dead cells, and you can really see also the correlation that these dead cells tend to also have a higher level of DNA damage. So this is particular assay that we could do to measure the level of DNA damage. And what we can see here is that the percentage populations, the samples that have more cell death, they also correlate with higher levels of DNA damage. And this goes back to also this point that some of these mixtures are a lot more toxic to cells than others. And we're still trying to understand why and how. And so one of the things that we have done in this study has been pretty exciting, has been to say, what is one quick thing that we can also do to complement our studies that is not just relying on expensive microscope and dyes and all these biochemically assays, but that are visual. We know that these morphologies get distorted. Can we quantify it? So we've been applying a lot of machine learning tools to really collect microscopic samples of all these collections of cells once exposed to a particular environmental type of pollutant. And what we have been doing is actually asking every one of those cells, how do I measure your overall shape? How do I measure your nuclear shape? And it turns out that there's a whole spectrum of how this is changing, but that they can actually group into very specific types. So then the question becomes, which ones of these are more susceptible to air pollution? Which ones of these are more susceptible to damage? And what drives that molecularly? So here's an example. Here's an example where we take one microscope data from all these cell samples, and every one of these little points represents a different type of an individual cell. So these are different types, and we can see these are all mapped onto this two-dimensional plot where we used about 33 parameters to sort of roughly outline how these cells are shaped, how their nucleotides are shaped. And what we see here in different colors are cells that are exposed to different conditions. So what you notice right away is that, condition pink here, whatever that PM sample looks like, tends to cluster around these shapes. There's a lot of yellow here that cluster these cells kind of here, and then there's some that are kind of in between. But we started noticing these extreme phenotypes here that connect and correlate with a specific type of exposure. So we can then cluster these and we can characterize these clusters. So overall, what you see here is that these are pretty healthy types of cells. And then you see this cluster 9 outliers here where cells start looking like they're dying. They're very small. Normally, we get pretty healthy looking cells like this. And then we start shifting to these really round, small phenotypes over here. So what you can see is that if you take control samples that we have not exposed to any pollutants, and we take these urban samples, which is our more drastic driver of a change in phenotype, what you can see is that these are very high in DNA damage and that they start clustering really here to these type of shapes, right? 9 and 10, these rounded small shapes. So this started to indicate to us, again, that not only were there specific mixtures and types that, we're still working to figure out what exactly is in them that's driving them, but that they had very specific phenotypes that we can actually identify. So then we started really focusing on understanding this connection between morphology of cells and back to what molecular drivers are resulting that are making given these phenotypes to these cells. And so I'll tell you a couple things that we've done that have been pretty interesting. So number one, I'm an RNA lab. We're really interested in RNA as a biomarker. So one of the early discovery that we have done is that consistently when we look at these mixtures, there's a lot of RNA oxidation. There's a lot of RNA oxidation, particularly of these type of modification, these 8-oxo-G, which is an oxidation of the guanine in the RNA, in the guanosine. And what you see is that consistently, when you have polluted air, there's a lot higher chances that we're going to see oxidized RNA. And what has been really interesting is that I won't bore you with the details, but we have tracked which RNAs get more propensity for oxidation. And there's some key pathways that are more vulnerable to others. We don't understand why. We still don't understand what properties about those RNAs make them more susceptible. But what is interesting is that they can encode for key enzyme, like one in cholesterol concentration. So you're probably going, if you're following me, this is starting to make sense because cholesterol, of course, is a key element in cytoskeletal properties and how cells get shaped. So there's an interesting connection here between these shapes, these compounds, their effect on pathways, their effect on RNA, enzymes, proteins, etc. So these are some technologies that we have developed to be able to get to some of findings like this. And in particular, this is something that we adapted and recently published. It's a method where we can extract total RNA and we can use antibodies that specifically recognize targeted modifications like these oxidations, and we can then do a pull down or kind of fish out those particular RNA types that are more prone for oxidation. So that's how we have figured out that there's a lot of RNA oxidation occurring here, and that there are some specific pathways that tend to be more prone and biased for this level of oxidation. And as I mentioned earlier, one of them that we have already identified is this cholesterol pathway. This particular RNA that encodes for these FDFT1 enzyme, which is a catalyst here in the cholesterol pathway, it's definitely one of the highest oxidized RNA. And when you assay cells, the intracellular level of cholesterol correlate pretty highly with the level of oxidation of this RNA that encodes these enzymes. So we're starting to really get excited about this understanding of, modification, especially because that is such a new field in really driving new phenotypes that are environmentally affected. If you are not as familiar with this work, I wanted to bring light to the fact that this oxidation, of course, is just one out of the many, many hundreds of modifications that people are really interested in studying in RNA as a new code that really affects cell behavior, and most exciting and important, that is affected by environmental effects. So there's hundreds that are out there being characterized every day by mass spectrometry and other methods. And a hot area in this field is understanding how do they arise from environmental stressors and what does it mean for cell function. And so another one that we have been really excited to identify in our samples is pseudouridine. So this is mass spec data from one of our collaborators. And what we see here is that the pseudouridine, other methylations, also accumulate pretty highly in abundance in some of our most exposed and vulnerable cell types that are exposed to these environmental sequences. So the second thing that we've been really interested in is, are they different patterns of gene expression? So we're also very interested in a systems view of all these pathways in the cells and how they respond to stressors. And so when we do these different exposures, we can collectively look at genes that pertain to very similar function in an analysis called a goal term analysis. And what we see in these functional clusters of genes is that there are things like cell migration and cell motility that, of course, contribute quite a lot to these phenotypes that we're seeing that are very correlated with exposure to these pollutants, to environmental exposure. So this is refreshing because, of course, as I showed you, there are markers that are being used that are more related to some of these other pathways that have been known for quite a while, like cytokines and whatnot. But this idea that a lot of the cell migration, cell motility, this is a new characteristic that it seems to be pretty sensitive that we can exploit here. And of course, it's very interesting to see that there are specific pathways and genes here that relate to ions, to metals out there, that it being triggered more by specific types of pollutant mixtures that we're seeing. So copper, cadmium, zinc. So it's starting to make sense as to why these things have differential effects. We're also interested in clonal populations. So I want to mention to you that a recent question that we're very excited about is, it's very interesting that similar populations have outliers. They are cells that are unique at being great at resisting these type of stressors. And they're cells that are super vulnerable. So at the single cell level, what creates this difference? Is there a program genetically that these cells already have where they're expressing genes from the get-go that quote unquote protect them versus others that make them more vulnerable? So we're using a lot of single cell sequence to actually isolate these individual patterns. So we have taken those clones that I showed you earlier, those really tiny round ones that were seen so vulnerable, so stressed, that exhibit a lot of toxicity, and some of the ones that don't, that really their shape remains the same, all indications genetically remain the same, and we are isolating those clones, reproducing them, so that we can create these individual clonal populations that we can study. And what we're seeing here is that indeed, when we look at these clones here on the x-axis and we look at the level of DNA damage, you can see the baseline here, even without any stressors. There is a big difference in how they are expressing a baseline set of genes that seems to really be conferring protection or extra sensitivity. And you can see that post-exposure, it just amplifies a lot more. So I'm not going to go through this. This is a lot, but what I want you to see visually, these are different genes So DOSP1, TRIB, these are all different genes. And what the colors here, these are different populations. The one to the left are unexposed cells. The one to the right are exposed. And what I want you to notice here is how these cells actually individually, these are different cell clusters. And if you look around, for example, here, you can see that these clusters here are super highly expressors of this type of gene, how these clusters here are, but not the other ones. So we're really excited in looking at these variations cell by cell to try to understand these concepts of biochemical, genetic susceptibility and resistance to stress. So lastly, what we have been also interested in is as engineers and people that are interested in working with communities and technology and making that available, what can this be all useful for at the end of the day? So I'm very excited to tell you that we have been working with collaborators that are experts in microfluidics to engineer new microfluidic devices with multiple channels where we can quickly test putting cells in there the effects of air pollution. And so we have our prototype that looks kind of like this. It's a little plate like this. And we can put multiple channels in our cells and really quickly use these indicators that we are developing where visually by microscopy, we can quickly assess the level of toxicity of these mixtures. And so this is very exciting because it really contributes to this vision of can we develop these high throughput systems to really assess these pollutants. And so we have a way to mix things and make high throughput mixtures. And then we have a way to link that to microscopy, and hopefully this will enable this vision. So in conclusion, I think I really wanted to tell you a little bit about my own interest and how I got to science. And this project is really dear to my heart because not only combines science and a lot of new methods that can move this very nascent field of RNA and RNA modifications forward, but is really a way to tackle from a science and a STEM perspective a social issue like, our environment and our pollutants and what are people breathing out there, and creatively even use technology to make that a conversation that everybody can participate with and have tools to really track. So we're excited about where this project is going. There's a lot of fundamental questions like why are some RNAs more susceptible than others? Why are these cells more susceptible than others? Can we transfer that from cells that are not and kind of engineer cells to be more resistant to really fight all these new environmental things that we're seeing? So we're working on this, but this has been a super exciting project. So with that, I just want to acknowledge some of the funding here and thank, of course, all the students that are participating in this project, our collaborators. This is a highly collaborative project. So I will stop sharing here and see if there's any questions.
Dr. Contreras, I have a question, if I may, quickly.
Absolutely. Nice to see you, Jacob.
Hi. Very nice, inspiring talk at the beginning. And I have a question in regards to the science. So 8OG and RNA, I apologize to kind of diverge a little. This as a lesion in DNA. Usually the repair DNA polymerase beta would look for this and fix that. Do you have any data or speculation on the impact of metals inhibiting that polymerase beta, specifically in these lung epithelial cells?
That's a great question, Jacob. So we have not looked at anything DNA related. They are those repair mechanisms. There's a lot of people actually looking at the effect of metals and thresholds for that can actually inhibit those processes. But we are very inspiring by your comment because the question is, in the RNA world, is it even an equivalent of enzymes and repair systems that work in similar ways to kind of police around these RNAs? Some people think this is a good thing. This might be a way to communicate to the cell that there's something extra that needs to happen. Why kill those oxidations? It could be a signaling piece. So there's a lot of debate as to what does this mean.
Okay, thank you.
I have a question too. Thank you for that fantastic talk. So my question, so I am Well, my research has a lot to do with cystic fibrosis, so I do care a lot about these lung diseases, whether they're genetic or not. Like so, cystic fibrosis, COPD, because my background is in infectious diseases. So I was wondering how your research on these how pollution affects these lung cells, um how does it connect to these kind of respiratory diseases like cystic fibrosis and stuff? Yeah.
Great question, Alex. Give me one second, Alex. My charger here is disconnecting. Give me 1 second. All right, my apologies, Alex. I just noticed I'm using a bad charger that is not charging my computer, so I changed it. So to your question, so what you're bringing up, this connection between all these diseases that are emerging, what sort of this environmental effects, there is a connection there that people have made based on the data that I showed you. You go to neighborhoods, you measure these things, you kind of notice this correlation. So for a while, it's been very correlational. And so one of the things that really inspired us to track this problem from a more molecular base is that there's a big mixing box there, right? A black box as to how do we understand these processes. So what you're asking about is extremely interesting. There's very little understanding of how these things are connected mechanistically yet, but a huge opportunity for us to understand. And I think what really excites me is that we've been working on RNAs, RNA modifications, epitranscriptomic things for many years, particularly on oxidation. But how these mechanistically work to end in diseases like what you're saying is very interesting. In fact, I will inspire you a little bit, I hope, by saying that NIH just released a call to actually for Alzheimer's, and it's very specific for studying RNA modifications in Alzheimer's. It's very specific for that. Any other questions?
Lydia, hey, I just wanted to ask you, I mean, I'm kind of blending your research with your passion for helping underserved communities and really changing society in some ways based on all of our scientific knowledge. So how long do you think this is going to filter through to actually change the way, our governments and our industry or cars or whatever really have so they can change and it can have a positive impact on human health?
That's a great point, Karen. I think that issues like this are really starting to catch the interest of a lot of people from different angles, from policy, from industry, from science, from medicine, from technology. And so one of the things I think a lot about that, and I have to tell you, it gets overwhelming when I think about how would this ever change worldwide. So what keeps me going on issues like that is to know that if I can bring light to this in a scientific way, and if I can do anything for communities to become more interested, even in tracking their own environment through science, that would be satisfying. And that's an achievable goal. That's something that wakes me up every day that I know could happen. So my vision that I can affect is at least use this problem, even when we go and talk to younger aspiring scientists, right? Middle schoolers, telling them about their social relevance, that their science can have an impact. It's something that we have to be doing. Because as I told you all from the beginning, for me, science was this very boring thing where you go into a lab and you move test tubes around. And it's like, what do you do with that? I don't want to do that. I want to be a doctor. Where people know what medical doctors do. They go every day, they help people. But we need to get better about, going and talking about problems that I think as scientists and STEM people, we have a lot to contribute to. So that's my goal number one. Goal number two is when I go and recruit students, grad students, that they see a connection in learning about RNA and RNA modifications and pull down and biochemistry and cell biology with something real that drives them every day and make them feel like they can have impact. And if we can achieve any of these goals, if we can deliver one, in this project, we delivered about 50 monitors to people's homes that didn't even know that science could do this. So we had to teach people how to use a monitor, how to monitor their house. We collected data from their houses. Kids were excited about tracking this every day, making graphs for us. So that was super exciting. And if we can even deliver one of these nano chips that we're making, that would be successful, Karen. So I hear you. There's a bigger problem out there. But I think it's so overwhelming when we think about changing the world that if we start with just very attainable goals, it just makes it a lot more fun to really think about.
And I just have to add here that I think that's why we're also dedicated to education for undergraduate students, graduate students, postdoc, is because we see them as really changing the world decades from now.
Yes, and not just changing the world, but changing STEM as a field, changing how we think, how we embrace each other, how we work with each other, the cultural attitudes about bringing your full self in it, being able to talk about your kids, about biking, about dancing, about hobbies. So I think there's a lot to look forward to, and I'm really grateful that you are having talented students like this come to this venue and really talk about these ideas. It's a lot bigger than just being in the lab.
Well, it's been our pleasure to have you today. Thank you so very much for sharing your story and your exciting and impactful research. It's been a real pleasure.
Thank you all. Have a great weekend, everyone.
Thank you. Bye.
Thank you.